Okay, so we've talked a little bit about molecular genetics, we talked about cytogenetics, we talked about Mendelian inheritance, and then overall the chromosome theory of inheritance, which puts all of those together. Now we're going to talk about some specific disorders like single gene disorders. Some rules to single gene disorders. First is they always follow Mendelian patterns of inheritance, which means you can use a Punnett square to determine the risk that the children's going to get it. You can do the same thing with a pedigree like you see over here. Pedigrees are basically, keep in mind, they're like adding Punnett squares together. So you can see one generation to the next, to the next, to the next, instead of just building lots of Punnett squares. So you can usually get it through a pedigree. All of these are based on proteins that are either structural or enzyme proteins that are dysfunctional. So structure or enzymes. And you're going to find between autosomal dominant, autosomal recessive that they help determine whether it's going to be a structure or an enzyme flawed. Okay. And then we'll talk about some of the X-linked. Right, so the first one, autosomal dominant disorders. First thing to remember with autosomal dominant is a dominant gene is the disease. So how many copies do you have to get the disease if it's dominant? You only need one. So autosomal dominant disorders, if a person's affected, that means they have at least one bad gene. They could have two, but focus on the one. Right? So it's pretty rare that two people with the same disease will actually have children. So somebody that has the same disease, like if they're an actually affected person with autosomal dominant, and when they pass it down to the next generation, it's kind of rare. But let's say that they do. So here's an affected parent here that's heterozygous, an affected parent here heterozygous. They both have the disease because they have the dominant gene. If they have offspring, then that offspring, 25%, will have homozygous for the dominant gene, which means they are completely have this disorder. 50% of them will be heterozygous. So do they have the disorder? Yes. It's a dominant gene. As long as that gene is there, they have the disorder. So 75% of their kids born will have the disease. 25% of their children overall will be homozygous, 50% heterozygous, and then this last 25% won't carry the gene, which means they don't have a disease and they're not a carrier. So when you look at these as they're being passed down generation, you'll notice that the characteristics of most autosomal dominant disorders are structural proteins, which means there's something wrong with the structures that they build, not enzymes, but the structures themselves. Things like achondroplasia or Huntington's that creates plaques or blockages in the brain. We'll talk about a couple of those. So first I want to talk about this recurrence risk. So I mentioned what's the odds of the kids getting it, and that's called recurrence risk. When you look at the recurrence risk of somebody with autosomal dominant, a key to keep in mind is that it's basically a 50% chance that they're definitely going to get it. Absolutely. So if it's just one parent, let's say dad's perfectly healthy, not a carrier, but mom has the disease. She has one bad gene. That means 50% of her kids are going to have it, period. In the last example I showed you if both parents were affected. But if just one parent's affected, then 50% of the kids are going to have it. So here's an example of that. Here you have mom has it, dad doesn't. Look at 50% of their kids have it. This person has it, 50% of his kids have it. So you can see, passing from generation to generation, about 50% of the offspring of somebody that's affected with an autosomal dominant disorder are going to have the disorder. Right? If they're a carrier, that still means they have the disorder. So here are a couple examples. First, achondroplasia. Achondroplasia, the name itself means without cartilage growth. Achondroplasia. It's the most common form of short-limb dwarfism. There are about 250 types of dwarfism out there, but this is the most common one. What's interesting about it is about 1 in 10,000 live births. It doesn't always have to be passed down from generation to generation in a hereditary fashion. It can actually be caused by mutation. So this exact same type of achondroplasia, about 80% of the cases are actually due to mutations. So random mutations, and it's because of the father. I know we give a lot of grief to a mother being over 35 years of age and leading to Down syndrome, but in this situation, the father's been carrying a sperm around for 40 years, hit with X-rays, ultraviolet light, toxins in the environment. You get mutations in the sperm. When spermatogenesis makes a new sperm and passes, he releases that sperm, that mutated sperm could fertilize an egg. And one of the risks of that is achondroplasia. So over the age of 40, there's a higher risk of achondroplasia because of the father. Another interesting thing to keep in mind, we know the different loci, we know the location, we know the gene that's involved in this type. It's a substitution of just one single amino acid, and it's glycine. One amino acid, that's all it takes. Remember, you saw this with hemoglobin and sickle cell anemia too. So it doesn't need a big difference. What's really interesting, if two parents are both achondroplasia and they have a child, if it's homozygous, so if mom passes it down and dad passes it down, and it's a double dominant gene, it's often lethal. It's almost always lethal. So the odds of a child being born homozygous for this disorder is extremely, extremely rare. So here's achondroplasia, some of the characteristics. The average height's under 4 foot 10 inches. The average male with achondroplasia is actually 4 feet 4. The average female is only 4 feet 1. You can see the pedigree, and in this example, it's passed down generation to generation, so it's not random mutations. This proband, remember proband is the first person, could have been a mutation, but everybody down past this is hereditary. So when you look at their trunks, the trunk's about average size. The head's usually a little bit enlarged compared to the trunk. The arms are shortened, and achondroplasia is actually kind of a misnomer. It says without cartilage growth, but in reality the cartilage grows. The problem is that the cartilage doesn't solidify, so it doesn't ossify and become firm bone. So these long bones, you know how the growth works with the epiphyseal plate, it's not lengthening appropriately. So they get shorter limbs typically. Some other things like health problems, when you look at the head, the growth of the bone in the head doesn't develop completely normally either. So you get a lot of times it'll squeeze on the auditory canal or the eustachian tube, and it causes a lot of ear infections. So a common problem that happens health-wise with people with achondroplasia is they get frequent ear infections. They can have respiratory issues. You know, they can't get a full breath, and a lot of times they'll get apnea. A lot of times they also get obesity, which is unrelated to the apnea, but would increase the problems with the apnea. One of the other risks you want to think about is the bones. Think of bones that encase things, like the vertebra. If the vertebra don't grow properly, then it compresses the spinal cord, and it's called spinal stenosis, and they get a lot of pain, tingling, weakness, difficulty walking. So those are some of the health issues. Otherwise, as far as intelligence, they usually have normal intelligence, unless there's something that's actually compressing the brain and causing problems with the brain. So those are some of the characteristics of achondroplasia, and of course know how it's passed down generation to generation. The next term I want to talk about is called expressivity. And expressivity is telling you that people with the disorder they don't always show the same degree of symptoms. So some people may have more extreme situations and some people less extreme. I'm still talking about autosomal dominant, but this can be in recessive disorders too. So expressivity is talking about things like, well, this is called polydactyly, too many digits. So here you can see an extra finger on the hand. Here you can see an extra toe on each foot. This is polydactyly. This is common in Amish communities, actually. And a lot of these diseases that are autosomal dominant or autosomal recessive, you'll see in communities, like ethnic communities, that prefer mating and having offspring with their own type. Like Ashkenazi Jews, they have kind of a clique, and they were kind of confined, of course, in Germany for a long time because of the wars. So they kept mating with people that already had existing disorders. They were carriers, so it increases the risk. Expressivity is talking about how bad is it. So you can have expressivity, something like polydactyly, where you have maybe one extra digit, and that's it. The other fingers and toes are fine. You can have a little bit more expression of it, where a finger or an extra finger and a finger or extra toe on a person, or you can have it where an extra finger and extra toes on every limb. So that's just an example of expressivity. Another disorder is called neurofibromatosis. And this is commonly referred to as von Recklinghausen disease. So neurofibromatosis, what happens is these neural tissues, so it can be gliomas, it can be neuromas, these things, they actually get firm, round, painful tumors that grow in the peripheral nervous system, grow out from Schwann cells typically. So they grow out and they can actually bulge out like you can see on this, or to an extreme situation like that. But expressivity says that you may get several little bumps, you may get a lot of large bumps, or you may just get these little things that we call port wine stains. That they're not extreme growths like this. You might have a couple bumps like that, but maybe have some dark structures on the skin instead. So that's an example of expressivity. Just because you have the gene for that disorder doesn't mean you're going to have this huge, life-threatening condition all the time. You may have mild situations. Another autosomal dominant disease is Huntington's, and we've talked about Huntington's once before, and we're going to talk about it again when we get to the nervous system. But for now, what you want to remember is it's autosomal dominant, which means that how many of the gene do you have to have the disease? Just one. What's interesting about Huntington's is that the signs and symptoms usually come out in the 30s and 40s, so they probably had children by now and not even known that they had Huntington's disease. Huntington's affects neurons in the brain that are inhibitory neurons. So if you're familiar with the terminology, they call them cholinergic and GABAergic neurons that inhibit movements. It looks a lot like Parkinson's, and we'll distinguish the differences more when we get to the nervous system. But what happens is it affects a part of the brain that's called the striatum. So down here in the core in the basal nuclei. And the striatum starts shrinking up. It also affects the frontal lobe. So those two things will tell you the symptoms. The striatum's there to help you control movements. So they get shaky movements. So they used to call it Huntington's chorea because it was almost like a dance that they would do and they would move. But it also affects the frontal lobe, and I hope you remember the frontal lobe's in control of things like personality. So they start going through personality changes at the same time, and that's Huntington's disease. Next thing you need to know about it is Huntington's. We know where the gene's at. We know the flaws. What's interesting is that it's a CAG repeat. So when you look at the DNA, if you see that CAG and it keeps repeating over and over like a broken record, if you have less than 26 of those repeats, that broken record skip, then you're normal. If you have between 29 and 36, then you're considered a carrier. You may not show symptoms, but your children are at risk because every generation gets more of those skips. Every time the DNA replicates, it gets more skips. So when you pass it on to your children, they will probably have 36 or more. Anywhere from 36 and more shows symptoms, so we call them affected. So this is an interesting disease, and that's a good example of expressivity. Every generation expresses the disease more than the one before. So if you have a Huntington's and you have a child that has the Huntington's, then it increases the risk. So think back. If two parents have a child, one of those parents has Huntington's disease, it's autosomal dominant. If you have it, you'll see it, but not until your 30s or 40s. And they have a child, what's the chance of the child having Huntington's? One affected parent, the other one's not affected at all and doesn't even carry. 50% risk. So anybody that's affected with Huntington's, their child has a 50% risk of having Huntington's. Right? And then you saw if two people with Huntington's have a child, then it goes up to 75% risk. So Huntington's disease, I put some overview here on this slide for you. It typically happens in the 30s and the 40s, and then it progresses for about 10 to 20 years until they die. Right? We know exactly what's going on. We know the flaws. We know that it's repeating CAG sequence. And then you can just kind of like scan through this. Whoops, that wasn't supposed to pop in there. And then I pointed this out a little bit earlier with achondroplasia, but it doesn't always have to be because of fertilization. It can actually be a flaw that happens because of a new mutation. So mutation can happen, and like I was talking about with mutation because of dad's sperm, achondroplasia is one of the problems. Progeria is actually another one. So progeria is kind of interesting. It's when the little kids look like they're a lot older than they actually are. So progeria is also one of those things that's passed down from dad to child because of mutation. It's not a hereditary thing. It's because of mutation. It's spontaneous happening. And what's interesting about progeria is it starts, you can see signs or symptoms of it about 18 to 24 months. They start aging a lot faster. Your immune system starts shutting down like an older person a lot faster. So you can see growth failure, loss of body fat and hair. They look like an older person. And then the problem is that they get a lot of heart diseases. So we consider it to be genetically lethal. All right. Marfans I mentioned earlier. Marfans, again, autosomal dominant. If you get the gene, you're going to carry the trait. You're going to actually have symptoms of it. And expressivity. Some people have all of the symptoms. Some people have some of the symptoms. But Marfans, what's interesting is that it's because of defective proteins. It's structural, which you should already know. It's autosomal dominant, so it's some kind of a protein structure. In this case, it affects things like heart valves. It affects the skin, the blood vessels, the joints, the eyes. You see some of the structures. Some of the symptoms, acrodactyly, which means basically it really means spider-like fingers. They look like they have long spider-like fingers. They look tall and lanky. Okay? A lot of times they have problems with vision because what's happening is that the tissues that are holding, the connective tissues holding the lens and everything in place are actually not working appropriately, so they have vision problems. What's really interesting about Marfans is they don't have decreased intelligence. In fact, one study found that they have a little bit higher than average. Their intelligence is about 110 points, which is above average. We think that one of our former presidents had it, and I think I spilled the beans earlier, but they think that Lincoln had it. So if Lincoln had lived longer, he probably would have had some more of these symptoms. And then now let's talk about some autosomal recessive disorders. Autosomal recessives typically affect some kind of an enzyme, right? Typically. Not always, but typically. So in this situation, the person that gets the disease, it's recessive. How many copies do they have to have? They have to have 2 copies of the bad gene. If they get 2 copies of the bad gene, then they get the disorder. If they only have 1 bad copy, then they're fine. In fact, we talked about this earlier, and you looked it up. When we talk about sickle cell anemia, just having 1 gene, 1 bad gene being a carrier is actually a benefit if you live in countries that have high risk of malaria because it prevents you from getting malaria. right? So some of the keys here, you have to have 2 bad genes. It's usually an enzyme that's deficient. Okay. Some of the common diseases or disorders are phenylketonuria, which we'll talk about, sickle cell, Tay-Sachs we mentioned earlier, cystic fibrosis we'll talk about, even color blindness. Right? So some forms of color blindness. So let me make that clear. There are actually over 20 kinds of color blindness. One we'll talk about, or we did talk a little bit about, that was X-linked, but other forms are actually autosomal recessive. So recurrence risk of AR. Well, look at these parents. So you have 2 parents. This one has the disease trait, this one doesn't. This one has the disease trait, this one doesn't. So do either of these parents, are they either affected? No. They have to have 2 bad genes. So what about 25% that become homozygous for a normal gene? Nope. They don't have it, they're not a carrier. 50% of their kids will be carriers, though. And then the homozygous affected in this situation is because they're homozygous, they have both bad genes and they have it. So about 25% of their kids are going to have the disorder. Right? If mom, just mom, is a carrier, she doesn't have the disease, how many of their children will have the disease? None of the kids will have the disease, right? Because all the kids would get a good, healthy gene from dad. But how many of the kids will be carriers because mom is a carrier? 50% will be carriers. Yeah. So you can kind of break that down and figure it out. It's interesting. I put a couple extra pictures in here since I sent you the slides. But you can see that when they mark this, they mark these are carriers. So this person doesn't have the disease, but it's a carrier. And then, again, 50% of the kids will be carriers. None of these kids have the disease. And since this guy's a carrier, 50% of his kids will have or be carriers. So you can see the carrier passageway. In this situation, you see a carrier met up with a carrier, and now about half of their kids are going to have the disease. Right? The next term to get familiar with is, well, with recurrence risk, is consanguinity. So autosomal recessive diseases, they really increase. Autosomal recessive is kind of interesting because they decrease in a population over time. But in populations that mate with people with similar genes, the risk goes up. So consanguinity is, in layperson's terms, kissing cousins. So consanguinity increases the risk because they carry similar ancestors' genes. So it goes up. So mating 2 individuals, I just looked at my picture of the people dancing the hoedown or whatever, and it just made me kind of giggle a little bit. But when these people will carry the gene in their family, of course it's going to be a higher risk that 2 people are going to pass it on to their child and have that actual autosomal recessive. Another time that you see it is actually in larger populations that are kind of isolated. Like certain Pakistani communities are isolated, and you see a lot of these autosomal recessive diseases that pass down from one family to the next. And this picture on the bottom is actually microencephalopathy, and it's something that happens a lot in Pakistan because this group was actually isolated. And because they were isolated, they started passing this gene around, so you see it once in a while. Another type of autosomal recessive would be PKU, phenylketonuria. And like I said before, enzyme deficiencies are what you really want to point a finger at. So it can be a catabolic enzyme, which breaks things down, or it can be an anabolic enzyme that builds things up. So I'll give you a couple examples. For a catabolic enzyme, think of phenylketonuria. If you had physiology with me, you already know what this is. But PKU is when you're missing an enzyme to break apart phenylalanine. So this list over here actually goes through it. You have phenylalanine, normal person. You have this phenylalanine hydroxylase, an enzyme that breaks apart phenylalanine into tyrosine. And tyrosine can be used for lots of things. You already know tyrosine can be manipulated and turned into hormones. Tyrosine can be used to build other proteins. It's a really handy or helpful chemical. But if you're missing this enzyme, phenylalanine accumulates in your body. And then it breaks down in a different way, but extremely slowly. So it turns into something called phenylacetate, which is actually kind of toxic and dangerous to your brain. So people that are missing this enzyme, they often get toxins built up in their brain that causes things like mental retardation. So that's one of the problems with it. Right? You can't give them the enzyme to break it down, so they just have to limit the protein in their diet. It's just a basic rule of thumb. But what happens, the phenylalanine builds up, it blocks transporters in the brain, leads to mental retardation. What's interesting is that some of the other characteristics of these people, they usually get more fair skin, and it's because tyrosine is also used to build things like melanin. So melanin is made from is a tyrosine derivative, or made from tyrosine. So if you can't make enough tyrosine out of phenylalanine, you can't make the pigments, which means they have lighter skin, usually fair eyes, like lighter blue eyes. They usually get musty breath. The musty breath is because that phenylacetate stuff builds up and it smells musty. A lot of times you'll see albinism. So the children can become albinos if it's really extreme, where they can't make pigments, period. So those are an example of catabolic. Anabolic means they can't build up. Cystic fibrosis, and I'll show you this in a couple slides again. Cystic fibrosis, you make a transporter. So this transporter enzyme is moving chloride, and they don't make it. So they can't make that. So let's talk about that next one. Cystic fibrosis. We know where the defects are. We know you can't transport chloride if you have the defect. So in other words, if you can't transport chloride, chloride builds up in all of the mucous membranes, and it holds sodium back there. When chloride and sodium hang out, you know by osmosis, water is attracted. So they get this thick, sticky, salty mucus. You should be able to figure this out. Where are the mucous membranes at? Mouth, respiratory passageway, urinary tract, reproductive tract, GI tract. So where are all the symptoms going to be? In all of those regions. They get thick, sticky mucus, so they have problems with breathing. The airways, the diameter of the airway gets smaller, so they can't breathe very well. Their digestive tract, when you're trying to excrete enzymes from the pancreas, it's thick and sticky, and you can't secrete enzymes, so they get things like malnutrition. Reproductive tract, they can't allow things to move, like the egg and the sperm don't move efficiently through the passageways. So you can figure some of these things out. Right? The next question you're going to answer is right here. So next homework question, number 7, is you're going to look up one of these diseases. It's called Ehlers-Danlos syndrome. It's an inherited disorder. It affects connective tissue. There are a lot of forms of it, but in all of the forms, what it does is it disrupts the collagen synthesis. So they can't make collagen efficiently. And yeah, this is actually a real picture of somebody with Ehlers-Danlos syndrome. So the connective tissue causes lots of problems, and I was about to explain it, but that's actually your job. So you're going to look up this disorder. There are 6 major types. It used to be 10, but now it's down to 6. So you're going to list off those major types. You're going to tell me the symptoms in general. All of them have similar symptoms. Just go through and list off the similar symptoms. You're going to tell me, is it primarily autosomal recessive, autosomal dominant, or is it primarily because of mutations? So when you list off the 6 types, put AR, AD, or mutation afterwards. Just tell me what it primarily is. Right? And then which of those types is the most common? Go ahead and pause and then come back when you're ready. All right. So the next term, special consideration, is something called penetrance. So penetrance, this is kind of like the lucky lottery. With penetrance, you could sorry, I had something that happened right there. With penetrance, you could get the bad gene, but you may not show it. Not the same as expressivity. You may get the bad gene, like for instance, retinoblastoma is a gene that you get that causes tumors in the eye. It's an autosomal dominant disorder, so if you get the gene, you should have tumors in the eye. But what they find is that not everybody does get the tumor in the eye. So they say that it's penetrance. Penetrance means that in that situation, only about 90% of the people will have something called incomplete penetrance, which means it doesn't always show up right away or it doesn't show up at all. Right? So when they talk about the penetrance of a disease, like they said 75% penetrance, that's the number of the people with a genotype that actually present with the disease. All right, and then the last group, single gene disorders that are sex-linked. So there are X and the Y chromosomes. The Y chromosome is for making a male, remember? So with the Y chromosome making a male, there are really only a handful of genes on there, and none that are super significant other than the SRY gene that tells us to be a boy. This makes me kind of giggle, but one of the genes that you really see a disorder with is hair growth. So hair growth on the ears is linked to the Y chromosome. It's called hypertrichosis pinnae oris. It means hairy ears. What's funny is it's on the Y chromosome, so if dad has it and passes on the Y chromosome to a boy, his boy's going to have it. So every boy that gets dad's Y with this disorder is going to get it. So it's kind of an interesting one. But most of these sex-linked jinx or traits are actually because of mom, right? So most disorders or I shouldn't say mom, because of the X, right? So mom can pass it down, but dad can pass it down too. I don't want to point fingers at either sex. I kind of said that inappropriately. It's primarily because of X-linked, right? So to get X-linked recessive means you have to get 2 bad X's if you're a girl or only 1 bad X if you're a boy. So you have to be homozygous female or a hemizygous male. Hemizygous means you only got 1 X chromosome and that 1 X is affected. Some of the common disorders with X-linked recessive are hemophilia A, Duchenne's muscular dystrophy, and Fragile X we'll actually talk about a little bit later too. So if you're a mother carrier, there's a 50% chance that your son will have the disease because he only gets an X from you, right? If you're a father that's a victim that actually is affected, I put victim, but an affected father, there's a 50% chance that you're going to give it to your daughter because you've got it on your X chromosome and you're handing it down to every one of your females. So if mom isn't a carrier, then that daughter is just going to be a carrier. She won't have the disorder. But father will never pass on this disease to his son because father will never give the son an X chromosome. Father only gives the son the Y chromosome. So it's kind of an important point to think about. And then next, hemophilia. So 9 out of 10 hemophiliacs actually have something called hemophilia type A. So that's the one we're going to focus on here. But they're missing a clotting factor. You can see the passageway here over to the side. All those different clotting factors are involved. It's like a chain of dominoes. If one of them doesn't work, the clotting process doesn't work. All right? So with hemophilia A, it's clotting factor 8 that's broken. With hemophilia B, it's clotting factor 9 that's broken. Hemophilia A is the most common one. Right? So hemophilia A, when they pass it down, it's missing one clotting factor, number 8. So if number 8 is broken, this whole chain of dominoes doesn't work. And here are some of the symptoms. They get easy bruising all over the place. If they break a capillary, they can't clot it to stop it from bleeding, so it actually bleeds under their skin. Here's another example around the knee. So if you fall on your knee and you break a bunch of capillaries there, that knee can actually swell gigantic because all of that bleeding into it. Right? And then here's a Punnett square where you can figure it out. So sometimes you'll see Punnett squares where they'll do the X and the Y's or XX, and then they'll put the little D afterwards for the disease, and that's how you watch it pass down. So mom is a carrier, right? And then remember, mom's a carrier, she's not affected because it's an autosomal recessive. And then when you pass it down, mom is going to have a girl carrier, but a boy with a disorder because the boy doesn't have a good X copy from dad because dad can't pass an X down to make up for that defective gene. So the next 2 slides I'm actually going to have you do on your own. This is a homework question. I'm going to have you look up Duchenne's muscular dystrophy. So muscular dystrophy causes a problem with something called dystrophin. And dystrophin is important because it helps hold the cell together. It holds the cytoskeleton to the cell membrane, so it gives support to the cell. If you don't have that, the cell membrane is weak. And if the cell membrane is weak, then calcium can leak in. And this is muscular dystrophy. So if calcium goes into a muscle, you know it's going to cause problems with stability of the muscle. So calcium comes in, it attracts water. Okay? It goes in and starts affecting the mitochondrial membrane. The mitochondria can't work appropriately. You get that process of vacuolation, which we talked about before. So you get vacuolation. You get excessive reactive oxygen species built up, which damages the membrane, damages the DNA, damages the protein cytoskeleton even further. That tissue starts going through necrosis, that cells will start dying, and then it replaces it with scar tissue, connective tissue, and adipose. So there's a quick pathophysiology of what happens. You're going to look up the background of it. So what are the symptoms? This is at a cellular level. What are the symptoms? How do they inherit this? So what's the form of inheritance? Is it autosomal recessive, autosomal dominant? Okay? Is it primary mutations? Right? What's the recurrence risk if the child has a mother that's a carrier? What's the recurrence risk if the child has a father that's affected, not a carrier but affected? Which kind of gave away number 1. Right? What's the incidence of the disorder? What's the gene and the locus that's affected? And what's the life expectancy? Go ahead and pause and then come back when you're ready. Okay. And then the next homework question, and the last for this section. So this homework question is basically over X-linked dominant. So you're going to look up X-linked dominant. Right? If the father's affected, there's 100% of the daughters are affected. Right? 0% of the sons are affected. Why is that? Figure that out. I mean, you know it because we kind of explained on another slide. If the mother's affected, 50% of all the children are affected. Kind of figure that out. I'm not asking those questions. You're going to figure that out on your own so you can look over here. You're going to look up one specific disease. It's called oral facial digital syndrome type 1. What's the gene name and the loci that's affected? So what's the gene name and the loci that's affected? You're going to look that up. Next thing you're going to look up is what's the incidence of this disease. And you should see a pattern. Before I told you that if you see something that's autosomal recessive or autosomal dominant, which is a single gene, compared to something that's complex inheritance, what are the odds of getting it? Are they going to be higher or lower risk? So you're going to look up the incidence of the disorder. You're going to look up the manifestations or the clinical features of the disorder. So what are the symptoms? What are the signs that this person has the disorder, outward signs? And then anything else you find that's interesting. So I put a little Punnett square here to help you figure out this part, and I put the pedigree here. These are the answers I'm looking for. This is your own, for your own benefit, but these are the answers that you're looking for. All right? Go ahead and do that. So pause the tape, and I'll see you on the next video.